The Kumar lab is interested in getting to know how different subunits that compose the N-methyl D-aspartate receptors, including the newer one that we have discovered recently in the lab, sculpt the functional properties of these receptors. We are also interested in deciphering the language neurons use to communicate with one another. We are also interested in getting to the nuts and bolts of temporal lobe epilepsy in order to explore new therapeutic avenues to combat temporal lobe epilepsy.
Our research focuses on the underlying pathology of what goes wrong in temporal lobe epilepsy and what the difference is between people who don't have epilepsy and people who do.
Mine is an electrophysiology lab. Electrophysiology is the mainstay of my lab. It allows you to record the electrical activity of neurons, and this equipment here behind me, it allows us to go to particular regions of the brain that we are interested in, impale these cells using a very famous and Nobel Prize winning technique called patch clamping, and allows us to record the electrical activity of these neurons.
One of the things I'm doing is I'm looking at this brain area called the paracebiculum. And what we're doing is we're looking at the frequency of activity. So how often is this neuron in the paracebiculum relating information to the entorhinal area, because that's what's connected, and we know the entorhinal area is a big area for epilepsy right now.
My lab has recently identified 7 different cell types in this region that's juxtaposed with the entorhinal area called the presubiculum. This was a structure that was not explored before and was thought to be homogeneous in its cell population. So what we did was we went and classified the types of neurons, electrophysiologically classified these neurons and correlate the structure of these neurons with function. To our surprise, we found 1 or 2 types of neurons, not all of them, that became hyperexcitable in these epileptic animals, and we believe that these are the neurons that actually drive the neurons in the medial entorhinal area to become hyperexcitable and eventually die.
The brain kind of works like a bundle of wires. So let's say each one of those wires has an on-off switch, and that's its 2 states. It can be on or off. And maybe normal function, we might turn on, 4 or 5 of these wires at a time, and that's gonna functionally communicate something to the region downstream. But in epilepsy, what's happening is we're just turning on hundreds of these switches at once. And this is just causing an overload of the circuit, and it just basically blows up. Well, right now the problem with temporal lobe epilepsy is that it's actually very hard to treat. It's often very resistant to medications, and sometimes in the most extreme cases, people have had to have the 2 hemispheres of their brain disconnected so that the seizures can't spread between the 2, or they have to have parts of their brain removed so that these seizures aren't occurring anymore. And these are obviously very invasive surgeries. There are very few parts of the brain that can actually regrow neurons. And that's why stem cells is a very big field right now, because the brain only creates a very limited number of new neurons, and these go to very specific regions of the brain. So if we can learn to replace some of these blown fuses with new stem cells, we might be able to find long-term lasting cures.
The goal here is to realize that these structures are heterogeneous. There are many different cell types and many different circuits, and by using electrophysiological techniques that we use in the lab, we are identifying specific neurons and specific circuits that might be responsible for driving these neurons in the entorhinal cortex in a way that makes them hyposynchronous and hyperexcitable, and eventually that is what leads to their death. We are very excited about the new type of NMDA receptors that we have discovered in the lab. These receptors, we believe, have very unique functional properties, and so we are very excited about exploring in greater detail the functioning of these receptors, perhaps in memory processes, perhaps in temporal lobe epilepsy. Their properties are unlike any of the NMDA receptors that have been described before. The immediate bearing of this research towards identification of these cells and circuits is that there are newer techniques out there like stem cell research and optogenetics, which rely on knowing which cells and circuits are involved in temporal lobe epilepsy, and perhaps down the line in the future we could utilize these techniques in a precise way to intervene in preventing seizures in animal models and also in human beings. This is a wonderful place to work. Florida State University is very unique in that it is not very much in the limelight, and I'm very fortunate to be working in this department, the Department of Biomedical Sciences, because there's an eclectic group of scientists here with diverse interests and backgrounds, and that has proved very beneficial to my growth academically. These days it's a privilege to be doing research, and the environment that the department and the college provides for me to do this kind of work is second to none. We are a small, lean, mean fighting machine. We have no dearth of ideas or questions or excitement for that matter.